Doing research is fun, but doing research with the facilities we have in the institute is awesome. We have all the state-of-the-art technology platform, and more importantly, this platform were managed by expert specialists and technical staff who are on top of the most advanced applications. So this together enable the top-notch research in the institute, and you will see why.
Cell biology is a science of art. It has been widely used to study the structure, function, and the mechanisms of all organisms at both cellular and subcellular levels. To achieve this goal, it needs a lot of unique and advanced facilities. The core lab of cell biology in IPMB has 3 divisions, including electron microscope, light microscope, and flow cytometers.
Electron microscope is a powerful tool for ultrastructural research. The electron microscope division was established in 1972.
The facilities are equipped with cryoscanning electron microscope, transmission electron microscope, and essential instruments for sample preparation. The scanning electron microscope can be used for sample observation by traditional and freezing methods. We have developed several key techniques for transmission electron microscopy, including ultramicrotomy, negative staining, shadow casting, freeze etching, freeze substitution, and immunolabeling. R&D of TEM technique has been focused on high-pressure freezing combined with freeze substitution and a set of sample preparing systems.
We provide a complete EM service, including sample preparation and instrument operation.
Every year, we hold a 5-day electron microscopy education and training session.
Light microscope division provides a wide range of microscopes, from conventional light microscopes to super-resolution confocal microscopes and image analysis resources.
This division has developed several microscopic techniques specialized to both plant and microbial samples. We provide high-quality and efficient services by using standard and advanced imaging techniques. For example, this isoriskin technique significantly improves spatial resolution and signal-to-noise ratio. Like a Stellaris 8-equipped hybrid detector and white light laser effectively eliminates the autofluorescence. Live cell imaging is conducted under a stable environment with well-controlled temperature, atmospheric gas and humidity, to ensure cells under observation are healthy and stable. Laser microdissection is a contact and contamination-free technique for the isolation of specific cells or area from section tissues. The captured cells can be used for quantitative and qualitative analyses such as PCR, transcriptome, or proteomics studies. The Zeislumer is a fluorescent stereo microscope, suitable for imaging large specimens in fluorescent and transmitted light for multi-channel, Z-stacked image acquisition. And with 12:1 zoom range and different illumination systems, to achieve the optimal imaging results of the samples. Using the TILES tool, we can acquire stacked images for the whole sample. High-resolution image acquisition allows for overviews of your samples with important details. Image J&E mirrors are used for 2D and 3D image analyses. We also provide on-site training and technical supports for both immunostaining and microscopic-related techniques.
IPMB flow cytometry core facility was established in 2010.
We are committed to providing high-quality analysis and sorting services for all researchers in academia Sinica. We also provide the most advanced flow cytometry expertise and research consultation. We have a Cytoflexus flow cytometer and a MoFlow XDP high-speed cell sorter. They can perform absolute cell counting, DNA and cell cycle analysis, and multicolor fluorescence analysis. Furthermore, high-purity four-way sorting and precise single-cell sorting can be carried out.
The facility is positioned as a comprehensive flow cytometry laboratory.
We have extensive experience in analysis of viruses, bacteria, algae, plant and animal cells, and will continue to provide cutting-edge research resources for cell biology.
Proteomics and metabolomics can be employed to profile proteins and metabolites in plants and microbes under different developmental stages or experimental conditions. Proteomics core and metabolomics core are mass spectrometry-based facilities providing state-of-the-art services to all the researchers in IPMB. Our mission is to use the cutting-edge proteomics and metabolomics technologies for deep understanding of molecular mechanisms in plants and microbes.
Currently, Proteomics core has 3 well-trained scientists and a high-resolution MS coupled with a new PLC for high-throughput proteomics analysis. We can handle a wide range of samples, ranging from microbes, cell lines, to plant tissues. Proteomics core offers a wide spectrum of services, including sample preparation for mass spec analysis, protein identification, identifying protein-protein interactions, identifying post-translational modifications, large-scale proteomic profiling, and targeted proteomics analysis. We can also access and operate the Orbitrap Fusion Lumos MS and Academia Sinica MS facilities for large-scale isobaric labeling experiments and glycopeptide identification. We provide preliminary data analysis, depending on the needs and capabilities of the users, by using proteomics software packages. Small Molecule Metabolomics core provides both untargeted and targeted metabolomic services for an unbiased screening of thousands of metabolites in a single run. The identification and the quantitative analysis of targeted metabolites in different organisms. We can access and operate the high-resolution MS Orbitrap Elite, located at Academia Sinica Metabolomics core facility for the detection and separation of metabolites. We develop customized metabolomics approaches according to the needs of specific projects. We also offer user training, workshops, and consultation of data analysis. For gas chromatography, the laboratory has mass spec coupled with automatic liquid and gas sampler for analyzing volatile organic compounds. For liquid chromatography, we have an ion chromatography coupled with sample collector for analyzing mono and oligosaccharide with high sensitivity. In addition, we have a new PLC using 1.8 micrometer column for services of high throughput, resolution, and sensitivity. The Genomic Technology Core has two divisions which provide services for genomic analysis, gene expression, and other associated technologies.
Welcome to the GTech Microarray and Sequencing Division. I'm Su Zhenzhou, in charge of this lab. The mission of our lab is to provide technical support and service to scientists in plant and microbial functional genomic study. Now we are providing array technology and NGS library construction service.
Our lab has a full set of equipment including hybridization oven, scanner, as well as gene spring software for data analysis for microarray experiments using Agilent DNA microarrays platform. We also offer RNA and DNA assays by using the Agilent bioanalyzer or fragment analyzer in order to check the quality of nucleic acid samples. The Droplet digital PCR system can be used to identify copy number variation, detect mRNA isoforms, and NGS library quantification. We have established and offered services for constructing standard next-generation sequencing library. We also collaborate with scientists to establish specialized protocols for different NGS applications. In the future, in addition to providing regular services such as NGS library construction, we will adapt or develop new technologies or experimental methods for genomic and transcriptomic research.
The DNA Analysis Division provides single sequencing for genotyping, sequence confirmation, validation of CRISPR-Cas9 mutagenesis.
For post-run analysis of sequence traces, we use peak trace software to increase the sequence read length to greater than 1200 pp. We also provide real-time quantitative PCR service for both relative and absolute quantification by using Applied Biosystems QuantStudio 12K Flex, 7500, and Step 1 Plus systems. The facility also maintains an eGene HAD-GT12 genetic analyzer for DNA separation and analysis. The goal of this division of GTech is to provide high-quality and cost-effective solutions for routine DNA sequencing and quantitative PCR.
In the post-genomic era, biological research uses imaging, mass spectrum, sequencing, and other experimental methods to attempt data. Database searches are also important for gathering information.
The mission of the Bioinformatics core is to use information technologies and the statistics methods to process various kinds of biology data.
In addition to using computing clusters, we are experienced in bioinformatics program development and integration, as well as statistical computation. Via working closely with biologists, we provide services that integrate biological knowledge, information technologies, and statistics methods. When handling a service request, we pay attention to definitions and computation procedures. We maintain reproducibility of our works by making work logs with detailed comments, as well as reviewing and reconfirmation. In recent years, we have been providing service items including genome assembly, genome variation calling, gene expression level computation, alternative splicing detection, alternative polyadenylation detection, co-expression network computation, customized gene ontology enrichment computation, biostatistics analysis and statistics consultation.